Hi guys, welcome to my YouTube channel. I'm Dr. Shuchita Verma and today I'm going to talk about another very important topic, that is serial dilution technique. Now the serial dilution technique is also known as viable plate count method. And why do we do this serial dilution technique in microbiology? It is one of the most important techniques in microbiology, because whenever you are dealing with microorganism, you need to dilute the sample. So why do we need to dilute the sample? Basically what happens is that microorganisms are everywhere, whether it is soil, whether it is air, or whether it is water. So you will find microorganisms everywhere and in so many large numbers. They exist in large numbers. So whenever, suppose we need to isolate microorganism or we need to enumerate the microorganism from a particular sample, let's take an example of a soil. So now, because soil has a large number of microorganisms, we need to isolate a particular microorganism. That is, the one microorganism is like pure culture. We call it as pure culture, where we have only one type of microorganism. Single species of microorganism is considered as pure culture. But how to get that pure culture? So there comes the need of this serial dilution technique, because if we take soil sample, then even if I am going to take 1 gram of soil sample, then also that particular 1 gram of soil sample has a very large number of microorganisms. But I want to get one particular type of microorganisms. So what I need to do? I need to reduce the number of microorganisms in my sample. So from so many number of microorganisms, how I'm going to get a lesser number of microorganisms, so that whenever I'm able to grow that particular microorganism in the petri dish, the kind of growth that I'll have will have the isolated colonies. Because now here the principle is that whenever we have any material or any sample that contains microorganisms, each microorganism, each viable organism, viable means living. So each living organism is able to form a particular colony. Now one viable organism will form one colony, right? So if we have few organisms, then we have few colony on our petri dish. But if we have a large number of microorganisms in our sample, then what is going to happen? We will have large number of colonies. And when we have large number of colonies, they will grow near each other, and then they might mix up with each other. So again we will get mixed population of microorganism. We will get mixed population, which we are not interested in. We are interested in having isolated colonies, because then we can pick that particular microorganism from the colony, and then again we can subculture it and preserve it and further use it for our experiments, whatever we want to do with that, right? So here comes the need of serial dilution, that what we are going to do is we are going to take the sample. It may be a soil sample, it may be an air sample, it may be an water sample, and then we will dilute it serially. One by one we will dilute it, make the dilutions, and depending upon the type of microorganism I want to isolate, I will choose the correct dilutions, right? So we have different set of dilutions to isolate bacteria, and we will use another set of dilutions to isolate actinomycetes and another set of dilutions to isolate fungi. So I'm going to tell you how to proceed further for making the serial dilutions. But before I start that, I need to give you some major steps or some precautions that you need to do, you need to take care while going for the serial dilution technique, right? So now, before performing any microbiological technique, you must take care of few things. Like, first thing is, the windows should not be open. They must not be open, because so as to avoid the air currents due to open windows. Then random hand movements must be avoided, because that will also create air currents. So random hand movements must be avoided. Another very important thing is that you must not talk while you are performing any of the techniques, because then our mouth has air, and then air again has microorganisms, and then you may end up contaminating the whole thing. So it's better not to talk when you are performing these techniques, right? That's very important. Another thing is that you must avoid any sort of contact between your sample and other sterilized objects like petri dishes and inoculation loops and everything whichever is sterilized, and you don't need to, and you must be very careful that it must not get contaminated. So you first should take enough precautions so as there is no contact between the sample, because ultimately your sample contains microorganisms and the sterilized objects are free of microorganisms, so there must not be any contact, right? So these are few precautions that you need to take, and now I'm proceeding with the serial dilution technique. So before proceeding for the protocol, I would like to mention here that depending upon the type of microorganism we need to isolate or enumerate, then we need to choose the type of media and also the dilution. So you need to take care that what dilutions are to be chosen to get a particular microorganism, and what is the type of media that you need to work with to get the right microorganism, right? So in case of bacteria, we need media that is nutrient agar. The choice of media is nutrient agar, and the dilutions which we use to isolate bacteria are 10 to the power minus 4 to 10 to the power minus 7. So now 10 to the power minus 4 to 10 to the power minus 7, then working with these dilutions, we will be able to have a comparative study that which dilution is giving the best results, which dilutions are giving the most appropriate isolated qualities, right? Then we have actinomyces. In case we are interested in actinomyces, then the choice of the media which we are going to use is glycerol yeast agar, and the dilutions used are 10 to the power minus 3 to 10 to the power minus 6. And with fungi, we will use Zappik agar media, that is CDA or Sabarod agar, and the dilution used are 10 to the power minus 2 to 10 to the power minus 5. Now the more the diluted the sample means the lesser the number of microorganisms and more isolated colonies, right? So now suppose let us take an example of a soil sample, right? So what am I going to do is that I am going to take 10 gram of soil sample, right? First I am going to take 10 gram of soil sample. Then I am going to take the diluent, that is 0.85% sodium chloride solution, that is saline solution. ठीक है ना? तो this is known as saline solution, that is 0.85% NaCl. This is our diluent in which we will mix 10 gram of soil sample. Now the point is that how much diluent we need, that is we will need 90 ml of 0.85% NaCl solution, right? Then 10 gram or 10 ml is equal to 10 ml, right? So 10 gram plus 90 ml, it makes up the volume to 100 ml, right? So this is how we are going to make our first dilution, right? Then from this we will further proceed and make the further dilutions. So 10 gram of soil sample in 90 ml of 0.85 NaCl, this will make our dilution 1 by 10 soil dilution. Now this is the 1 by 10 soil dilution, right? In case of in place of this saline solution, you can also take water blanks. You can also use sterile water in place of these saline solution, you can also use sterile water, right? So this is our first dilution, and then what I am going to do is, from this I am going to take 1 ml aliquot and mix it in 9 ml of water blank, right? Water blank or saline solution, both you can choose among those. Either you go with this saline solution or you can use sterile water. So consider that I have taken these test tubes and I have added 9 ml of saline solution to these test tubes, right? And I am going to label them as 10 to the power minus 2, 10 to the power minus 3, 10 to the power minus 4, 10 to the power minus 5, and 10 to the power minus 6, right? So from this I am going to take 1 ml aliquot and add in this water blank, right? So the whole volume becomes 10 ml. 1 ml of aliquot plus 9 ml of the solution. So it becomes 10 ml. So this is my dilution and I am going to name it as 10 to the power minus 2, right? Now from this test tube, I am going to take 1 ml with the help of a pipette. Aseptically, I am going to transfer this 1 ml into this test tube. Now this test tube already has 9 ml of this saline solution, 0.85% saline solution. So now this dilution is 10 to the power minus 3. I am going to shake it well and then aseptically from a fresh pipette, I am going to pipette out 1 ml from this tube and add to 9 ml of this blank. Then 1 ml plus 9 ml, the total volume becomes 10 ml and the dilution comes to be 10 to the power minus 4. Similarly, I am going to take another 1 ml from this, add to this one, and now this becomes 10 to the power minus 5. And I am going to repeat it for this tube as well, and this is my dilution 10 to the power minus 6. Further, I can make such more dilutions like 10 to the power minus 7, minus 8 and so on, as much as I want, right? So this is how we dilute our sample. So these are our media plates, right? These petri dishes have already solidified media. So now say consider this dilution 10 to the power minus 2. I am going to shake it well with the help of the pipette. I am going to micropipette. I am going to take 0.1 ml of the aliquot from this dilution and add to this media plate, right? Similarly, 0.1 ml of aliquot from this particular dilution into this media plate and label it as 10 to the power minus 3. Now label it as 10 to the power minus 2. This petri dish has 10 to the power minus 2. This petri dish has 10 to the power minus 3. Similarly, again 0.1 ml of the quantity of this aliquot I am going to add to this petri dish and label it as 10 to the power minus 4. You must label all your petri dishes in advance, and while taking care that you need to add 0.1 ml of the particular dilution, the particular aliquot into the properly labeled petri dishes. To save the time and to save the extra effort, you must label all your petri dishes with the time, with the date, the type of media, the type of dilution you are using, and every detail in advance only. I am telling you here just to make the process easier. Otherwise, you must label prior to your experiment, right? So now this is the petri dish for 10 to the power minus 4 dilution, and I have taken 0.1 ml of this and added to this petri dish. Similarly, from 10 to the power minus 5 dilution, I am again going to take 0.1 ml aliquot and add to this particular petri dish, label it as 10 to the power minus 5, and then incubate it. For the desired time at a particular temperature. Like in case of bacteria, we incubate at 37 degrees Celsius for 2 to 3 days, right? And in case of fungi, we incubate at the temperature of 25 degrees Celsius for 7 days. Following which, we will get the colonies on the petri dishes. Now, with the help of the colony counter, you can count these colonies. With the help of the colony counter, you can count these colonies, and there is a very important formula that you need to apply to calculate the colony forming units in the sample, right? So, this is how we proceed, and now I am going to show you the calculation that how to calculate the CFU, right? Now, how will you calculate the number of viable cells per gram or ml of the given sample, right? So, number of viable cells. Again, we will move back to the principle. The principle is that any material containing microorganism. Now, any viable cell will be able to produce a colony. Non-viable cell, that means non-living, will not be able to grow into a colony, to form a colony. No. So, we will only be able to calculate the viable cells here, right? So, viable cells per gram or ml of the sample will be equal to mean plate count. Now, when we were doing the experiment, we saw that we have inoculated 10 to the power minus 2 labeled petri dish with 0.1 ml of the sample, right? But there was only one petri dish that I showed you in the diagram. But in reality, when we perform an experiment, we need to do in triplicates. Triplicates means we need to do in triplicates. Triplicates. So here the word is tri, means 3. So for every dilution, you need to put 3 plates, right? So now let's say in plate number 1, you have 10 colonies. In plate number 2, you have 15 colonies. And in plate number 3, you have 25 colonies. Just for the dilution 10 to the power minus 2, right? So what you are going to take, you will be going to add all these. You will be going to take the mean of this. So 10 plus 15 plus 25 divided by 3. This will give you the mean. Let's say x. Okay, so here you need to take the mean of these. Mean of these plate counts, right? So for every dilution, like we are using 10 to the power minus 2, 3 plates. Then dilution 10 to the power minus 3, 3 plates. Then dilution 10 to the power minus 4, 3 plates. Then dilution 10 to the power minus 6, 3 plates, right? And so on. So for all the triplicates, you need to get the mean plate count. That is why it is written mean plate count. सिर्फ प्लेट काउंट नहीं लिखा है, मीन लिखा है. तीनों का कंसिडर करेंगे. मल्टीप्लाइड बाय द डाइल्यूशन फैक्टर. नाउ हमने डाइल्यूशंस बनाए थे, बट डाइल्यूशन फैक्टर we didn't find this term. What is dilution factor? Dilution factor, what should I write? Dilution factor is the reciprocal. It is the reciprocal of the dilution. Now, if my dilution is 10 to the power minus 2, then the dilution factor will be 10 to the power 2. If my dilution is 10 to the power minus 3, then the dilution factor will be 10 to the power 3. So, I am going to multiply mean plate count by the dilution factor divided by the dry weight of the soil. So, before proceeding, you first need to take this dry weight of the soil, whatever amount you are using. If you are using 10 gram, then take 10 gram, right? So, this is all about serial dilution technique or the viable plate count method. and I hope that you must have understood the concept nicely. In case you have any query, just leave a comment in the comment section. In case you have understood the concept, do let me know that also in the comment section, because I'll be the happiest person to know that you are able to get the concept nicely and I'm able to explain you nicely. So that makes a huge difference in my life. And then don't forget to share this video with your friends and anybody who is interested in microbiology people. Do like and share the video, right? That is all for today. Have a nice day. Bye.